My name is Roger Barthelson. I'm talking to you from iPlant. We're going to talk about transcriptome assembly today. Okay. It may seem like a trivial question, but it isn't entirely. It's not a common English word. Transcriptome. It can be defined as all the RNA molecules in a cell, but when we're looking at a transcriptome, we're discussing a population of RNA molecules, and we try to develop a representative population or a representative set of sequences that represent well that population. And that population may be for a single cell, typically a tissue or a number of different related cell types that can be dissected from tissue, cultured cells, or it may actually represent the transcriptome of a whole organism. It depends on what you are studying, and even though it says here that it includes ribosomal RNA, tRNA, other non-coding RNAs. Typically, the small RNAs are not going to show up in a lot of the protocols commonly used, unless you choose a protocol that looks for them specifically. And so a lot of the time, we're really looking for the messenger RNAs. And to start off, we're often focused mostly on proteins. So that's sort of the emphasis we're working from today, although a lot of the methodology is similar. Now, doing transcriptome studies is typically part of RNA-seq studies. And for doing RNA-seq, we're typically studying transcribed RNA, as I said. In order to do that, we're looking at fragmented RNA, so that you understand that we're not looking at RNA that's synthesized or a representative cDNA that is synthesized from a specific site that can be recognized and defined. It's not just the poly-A RNA, reverse transcribed. It is the RNA that may be reverse transcribed from poly-A, but it is also fragmented, so that you're looking at random sequences. So, and that's key to the whole methodology. You're really putting your RNA through a shredder. It's like, as this reference describes, it's like putting a picture through a shredder and then trying to piece it back together. Thank you, Abigail, for this this great cartoon that I stole from the internet. But I thought it was a good representation of what's going on. Today we're going to focus on the transcriptome assembly part. But what's first, if any of you were listening last time, I made a strong point, I hope, to emphasize the importance of read cleanup. Because whenever you're doing an assembly, if you don't have clean sequences to start with, you're going to be assembling junk to junk, as well as your actual sequences that you're interested in, assembling them together or assembling them to junk. So you're going to make a mess to some extent. It may look, to start like an assembly but it's not going to be nearly as good an assembly if you don't get rid of the junk. And that's why I started out by talking about read cleanup last time. And that webinar is recorded. It's in the can, as they say, so you can look it up and view that if you need to. But we have some really great protocols in the discovery environment at iPlant for doing read cleanup. So what? After that, I mean, the transcriptome assembly itself, are we talking about reference-based assembly or de novo assembly? That is the question for you, the first question, because you theoretically know what you're working with. You either have, you are either working with a reference sequence or a reference organism, humans, Arabidopsis, Drosophila, something that has been sequenced before, has a genome sequence at least, and so you can use the reference to help guide your assembly. But we're going to focus on de novo assembly today, because that's often what a lot of you will be facing. And you know, a lot of the principles can be the same. A lot of the questions that come up may be the same, but they're not entirely. It's not entirely the same problem. Reference-based assembly is theoretically easier, but it you know, it has its own concerns. So for doing, we're doing this discussion today, we're going to focus on Soapdenovo-Trans. It's one of a series of assemblers from BGI, and those assemblers, Soapdenovo, Soapdenovo-2, and now Soapdenovo-Trans for transcripts. They've all evolved, and they can be pretty good assemblers. They can do a pretty good job. And as you may know, BGI does a lot of work with Illumina sequences. And so it's designed for use with Illumina sequences, and most of you will probably be working with Illumina sequences, and that's why I chose it today. But feel free, if you have questions at some point later on about other assemblers or for larger reads, for example. We have those at iPlant 2 and available through the discovery environment. They're just not used nearly as often. So, as I said, Soapdenovo-Trans, as the name implies, is a transcriptome assembler, and it's de novo. And one of the interesting points that I wanted to make today is that a lot of you come to this webinar with the words or the word Trinity on your lips, because that's what you think of first, many of you, when you think of a transcriptome assembler. And Trinity is a good assembler. I hesitate to be critical at all, but Trinity does have problems in terms of how it works with large computing systems and the amount of time it takes to run. So this table sort of summarizes pretty well the differences between Soapdenovo-Trans, Trinity, and a third assembler, transcriptome assembler, that's available through the discovery environment, and how Trinity by far is the biggest hog of time. And that can be problematic. It can be problematic for us because it uses a lot of our user time on exceed systems, which are the supercomputing systems that are available for all scientists' use in the United States. And so there's limited time available. And you have to be careful about how you use that time. So, it's not that you can't use Trinity, it's just that think about using Soapdenovo-Trans. And I'll give you some more information about that later on when we look at how to evaluate our transcript assemblies. So when piecing our transcriptomes back together after we've shredded the sequences randomly, and we have this set of randomly distributed reads, or hopefully randomly distributed sequencing reads, coming from the whole transcriptome, from transcribed RNA from all over the genome, everything that's transcribed within that tissue or within that organism, depending on how you capture that information. Typically, though, if you're looking at a representative sample, you're looking at a single tissue, maybe a mixed cell type population, but you're not, but sometimes you may, at least for smaller organisms, grinding up the whole organism and getting RNA out of the whole thing. So, but again, keep in mind, whatever you started with in the blender is going to be represented in the RNA population that you're looking at. And I'm not going to spend a lot of time on this, but I assume that many of you have heard of the use of K-mers by a lot of the modern, in a sense, assemblers, and the Brun graphs that are part of the reassembly process. And K-mers being the defined length sampling that we use, say 25 mer, and read that 25 mer reading from one base and then from the next base and the next base, so that we have a total, in a sense, very redundant sampling of all your sequencing reads. So you end up with a lot of K-mers, a really big K-mer population, and then you're looking at how those K-mers can relate to each other and be reassembled using the De Bruyn graphs, which I'm not going to spend a lot of time explaining because I'm certainly not an expert on them. And it's still sort of early for me, even if I've had 2 cups of coffee. But it is important to understand K-mers in the sense of when you're choosing a K-mer value for Soapdenovo-Trans. And as I recall, that's not something that you do for Trinity. I could be wrong about that. But many assemblers allow you or require that you choose a K-mer size. And you have to sort of balance 2 considerations. One is that if you use longer K-mer value, then you will get maybe a more accurate sampling of, that is, when you take a Kamer from an interesting piece of sequence, and it's a large Kamer, then you're covering more of the sequence, and it becomes more unique to that piece of sequence. On the other hand, if you take a shorter one, you don't have that advantage, but you can get a lot more Kamers out of the same population of sequences, and so that has a value in itself, because it means that you can get a better sampling of the reads, the sequencing reads that are, or sequences that are well represented within those sequencing reads. Let's put it that way. And so, summarize, larger K means it's more specific, and a smaller K is more sensitive. So to some extent, it's something that you have to decide. Am I more interested in getting the low expressed members of the RNA population and learning what they're doing? Or are you more interested in getting a more accurate accounting for what's going on with the main population of RNA molecules? It's not that they're not both important, but one may be more important to you than another. And you also have to just keep in mind that there's a realistic level of sensitivity that you can achieve with a given amount of sequencing reads. So sometimes larger Kamer settings are suggested, and that can work well, but often, some of these Kamer values that are shown here are pretty large, and not necessarily, if for a smaller sampling, not necessarily the best choice. You may well find that you'll get better assemblies if you use lower Kamer values. And this is something that you'll have to gain some experience with. But I often find for transcript assembly that you're going to do as well in the range of 25 to 31, and you will notice that we tend to use odd numbers, and there's a mathematical reason for that as far as Kamer selection goes, but I'm not going to explain it to you because I'm not a mathematics major. And it's not necessarily
Roger? Roger, did you mute yourself?
Lost audio.
Yes, that's right.
Okay.
Yeah, I'm back. My foot is in a knot of power cord, so I just tripped the switch, I think. But I'm back. So when we're looking at oh, you're getting an echo? Is everybody else out there getting an echo?
No.
No echo for me.
Okay.
Who's getting the echo?
Uwe.
I'm good too.
If anyone is getting an echo, could you put it in the chat? Just the yeses?
Yeah, I'm not sure where that would be coming
Yeah,
from.
me neither. Okay, Uwe says it's okay now.
Okay. Okay, so if you're experiencing an echo, I'm sorry. but I'm not trying to echo. um, So one of the things that you get once you've done a Soapdenovo-Trans assembly is some files like the SCAF-seq file for assembled transcripts. You get a really helpful SCAF statistics file that gives a lot of great statistics, so you don't have to do much to analyze immediately or get a sense of how the assembly went. And you get some other useful files. But before we go into that any further, does anybody have any questions at this point?
You can either put questions in the chat window or unmute and just speak up.
Yeah, it's up to you. And there will be an opportunity at the end to ask questions also.
Roger, there's one in the chat window from Meg Allen. It says, how big are the files and what do you need to open them?
In the discovery environment, nothing. All you have to do is click on a file and you can read it in the discovery environment, any file. Even a binary file, even though most of us can't read binary, you can look at binary in the discovery environment. And that's what I was going to show you right now.
Any other questions from anyone? So, Roger, do you have your chat window open?
I don't.
There's another question in there. I can either read it to you.
Let me see. What input files are required for the assembly? Are they the same as for Trinity? Well, it's whenever you're doing an assembly, you have your reads from your sequencing. You've done the cleanup, and so whatever your reads are, those are your input files. And it's different assemblers will allow you to input the reads in a different fashion, but it's all about the reads because the assembler doesn't need any other files. If you're using Soapdenovo manually, typically you may have to, you may decide that you want to write out a config file. But in the discovery environment, we take some pains to create a GUI interface for these command line programs. So the information that would typically go into a config file is captured in the app window and then sent. It's literally written into a config file and used to run your assembly. And I'll show you that right now.
Okay, Roger, do you want to hold off on Zane's question until later?
How do you calculate the insert size? Well, it really should. Whoever prepared the samples, if you have paired end sequencing, if you're only doing single-ended sequencing, then you don't have an insert size. But if you do have paired ends, then it's really up to you to find out. Or the sequencing company, if they prepared the libraries before sequencing, they will know what the insert size is. If they can't tell you, then maybe you should use another sequencing company because it's part of the preparation. Often you will tell them specifically yourself. Okay, and there's a question on using a range of k-mers and merging. Yes, you can use multiple k-mer values. I think you'll find that that's a diminishing value. I mean, I think it is helpful for you if you haven't done assembly before with a typical experiment to try a series of different k-mer sizes and see how the assemblies work out. But I think you'll often find that that you hit a range pretty quickly that where it doesn't really make a huge amount of difference. But it's important to understand what that range is for your set of, for your length of reads and the size of population of reads that you may be working with. And so trying multiple k-mer values can be helpful for that. And that at least is an option for Soapdenovo-Trans. It's pretty hard to do with, well, again, I don't think Trinity uses a k-mer setting, so you don't have that opportunity anyway. I think it does its own routine. And so Zane points out that it's the best way to determine insert size is really to map to a reference of some sort. But that only works if you have a reference. If you're doing de novo assembly, often that means you don't have a reference. And that's the problem. Otherwise, there are ways of doing it by mapping. But if you don't have a reference to map to, then you can't use that approach. You are, and the insert size is literally defined in typical sequencing procedure by the size of the DNA that is cloned or amplified in a population. That is, you're looking at reads from one side to another for paired end sequencing. And so even though if they're randomly distributed, you're still limited to reading from the ends, and those ends are spaced by the insert size. And so it's literally based on what size of DNA was cut out of a gel, or in some cases, what size was your RNA, your set of RNA fragments, depending on the procedure that you're using. It's been a few years since I've done an Illumina procedure, and I know they've evolved a bit, but it comes down to essentially that. So as I said, somebody knew when they were doing it what the size was. And whoever prepared the sequences should be able to answer that question.
Okay, so great questions and discussion, Marissa. Thanks for chiming in, because that's something we do want you all to do, is you all have a lot of knowledge too. Roger, why don't you go ahead and move on, since we're at 30 minutes after the hour now.
Yep. Okay. So I wanted to point out that Soapdenovo-Trans is found down in the high performance computing section. And it may be sort of confusing to look at my collection of applications, because you'll see doubles and multiples of them, because I built many of these, and so there's my personal copy and then the public version that was made from that. But what you will see is that, what you'll commonly see is is in this section, you'll see the all of the assemblers pretty much represent. And let me make sure I'm picking the right one. I forgot that this can be confusing when I want to look at the public version. But I think it's this one. It should pretty much look like this one in any case. And the key is that I gave you an option of 2 paired end libraries as inputs. I could have put in more. If it ever becomes a problem for you that for some reason you use more than 1 or more than 2 different paired end spacings with your transcriptome studies, let me know and I'll add it to my list to do sometime, but I certainly won't have time to do it immediately. So, and there's 1 input for single reads because all single-ended reads are pretty much the same and you can concatenate them all together before you do an assembly. And then you have the option of putting in, or the requirement in some cases, a number of different parameters. One parameter that you absolutely need is the maximum read length. Anything with a red star is required, and you can't start it without putting in a value there. And the Kamer size needs to be set also. The rest is up to you. And to some extent, it will go along with matching your input data. There will be explanations for these and what is required if you, or what it actually means to some extent. That is, for read orientations, typically you're just going to leave it at 0, for example, but if you, for some reason you did mate pair sequencing then you would need to put a 1 in that space. But most of the time, and that's why it's not required, it's just left as 0. Usually you're going to do all the assembly steps for the paired end libraries, and just the first assembly step, which is to assemble contigs, because you can't do scaffolding, which is the other assembly step with single-ended reads. And you also should put in your insert size. And if you don't know, then I would just guess. If that's the best you can do, put in 500, and that's a good likely guess. And so that's how you set up, in a nutshell, a Soapdenovo-Trans. Once you've started it, the job is submitted to a server at TAC. That's the Texas Advanced Computing Center in Austin, Texas. It's an Exceed Center, and it's a really big computing system. So it may take a little time for it to get through the queue on the machine before the job actually runs. But when it's done, the results will get back to you just like any discovery environment job. And I also wanted to point out that in the community data section of the data store, if you look at iPlant training, there are a lot of things, but there's somewhere in here, okay, transcriptome assembly. So there's a transcriptome assembly example, and you can look at some of the, for example, sample input data if you want to try your hand at it. And this, these reads are essentially the same reads that we used for read cleanup last time. So if you were part of that webinar, then you, or if you want to go back and look at it, you can use the same data straight out of the HT process, Trim-O-Matic, and use it with Soapdenovo-Trans. And then you get your output from Soapdenovo-Trans, and there are a lot of files, as said, and SCAFseq is the one I often look at first because that's your scaffold sequence. And as you see, all you have to do is click on a file and you can see. And you can see that some of the scaffolds are really nice and big and some of them aren't so big. And these end spaces in there, well, that's because it's a scaffold. There are contigs that are pieced together using the information it has on the spacing from the insert size. And so that's why you need that information.
Roger?
Yes.
There's a question from AGI in the chat. It says, do you want to wait until you're finished with what you're doing? You can normalize the reads. I don't, and sometimes that helps, especially if you're dealing with a lot of data. It can be very helpful to normalize the data because if nothing else, it reduces the volume of data that you're trying to feed into an assembler. And assemblers do have limits. If your Trinity runs or your Soapdenovo-Trans runs even are running too long to fit it within the 48-hour limit of the Stampede server where they run at TAC, then you want to look at reducing the size of the wad of reads that you're trying to stuff into that assembler. The assembler doesn't work well if it's overloaded. And that's why you need to do that, but also because you'll never get any results if it runs too. And there are apps available like Trinity Normalize, K-mer coverage. And I'm not going to spend time talking about that today, but that is, it's designed to work with Trinity, but you can use it with Soapdenovo-Trans, and you may see some improvement. I don't remember seeing an awful lot of improvement with it. And if you read on the website for Soapdenovo-Trans, you may see information that talks about all the stages. And I can't remember, there may be a normalization built in. But there are different normalizations and read correction routines used by different assemblers, and sometimes they can be helpful. Anyway, I think I'll go back to the presentation. And at this point, after you've run your assembly, typically you're going to be interested in the N50 statistic, or something else that's important, the core gene representation. Those are two ways of evaluating your assembly. And this is a simple explanation of what an N50 represents. Many of you have seen this before, but it's helpful to think about it again. And again, these slides will be available so that you can go over them in more detail if you need to, or it can be helpful when you're thinking about these things. But one of the, and as I showed you before, there's .scafseq
or .scaf statistics file is quite helpful because it does provide an N50 statistic. It provides some other statistics as well. But these are essentially statistics. They talk about, well, how big typically are your transcripts that are assembled. And that's an indication of maybe how successful the assembly is, but it doesn't really give you any information on the accuracy. Whereas SEGMA, or KEGMA, depending on how you like to pronounce it, gives you an indication of actually the completeness of the assembly. And that's one of the reasons I like using it. And so it goes through a routine for determining what are the apparent core peptides represented in your population of transcripts. Core peptides being what's defined by this package as KEGs or SEGs, core eukaryotic genes. And so it's defined those broadly for eukaryotes because based on what's typically seen with eukaryotes. And it has a set of, I think, 244 different ones that are considered core genes. And it allows you to analyze your transcriptome based on that. And I'll show you an example of that in a second. But in general, otherwise, when you're looking at, you're evaluating your transcriptome, if you feel like at this point that it's a good assembly or a pretty good assembly that you want to work with, perfection is not going to be found. But if you feel that you've done roughly as well as you're going to in a reasonable amount of time, then you may also want to take a second and run CD hit EST, which clusters together some similar transcripts in case some of them are just slightly different because of an error in sequencing. And then there's also transcript decoder, which will transcribe them into peptide sequences. And that will give you a sense, ultimately, of what are the actual coding sequences, which is useful because if you're looking at a population of transcripts, you expect to see maybe mostly coding sequences in there, and so how much is left once you take out once you've eliminated those that aren't coding sequences, gives you a rough indication of how good your assembly is too. And then you can go on and do annotation. And that typically may require doing a BLAST P with your peptides that you created with transcript decoder. And I would generally recommend using UniProt database or a selected population of that, because that ties directly into your gene ontology analysis. And it's a reasonably compact set of peptides for comparison. But it's still going to take you a lot of time if you try to use the whole UniProt database. But we don't have a lot of time to talk about that today. the emphasis is on the assembly here today. So one of the things I wanted to go over is that for the Belgica antarctica reeds, this is for my favorite insect, or one of my favorite insects. It's from Antarctica. It's a midge. It's wingless, and it has a really small genome. The reads are available in the SRA database, and that's what we used, as I recall, for the read cleanup webinar. And if you feed those into a typical assembly for Soapdenovo-Trans, these are the results you get. N50 of 2781, roughly 30,000 for the longest scaffold, and the sigma score for full sets. The core genes are divided into representative types of genes or of peptides, and roughly 97% of them are full. That is, all the members of that particular family are represented in this population, and this is by looking at essentially comparing the sequences. But 99% of them are partial families. So a lot of, most of the genome seems to be pretty well covered. Most of the transcriptome seems to be pretty well covered. And for Trinity, you can say the same thing. The numbers are a little lower for these scores, a little higher for this score, and that's an impressive difference. But keep in mind, they use different approaches in the end. Is this a really important thing to have long transcripts? Maybe it is for you. But on the other hand, are they representative of a complete population? Well, they are pretty well representative. They're very similar scores here. But Soapdenovo-Trans takes a lot less time, takes a lot fewer resources, and it may really be a better assembly. The only way you know is by studying further at this point, because by your quality and completeness analysis, they look pretty much the same. And Trinity produces a lot of files, but are they good files? That's a reasonable question in itself. Another thing I just wanted to point out was that for the transcript decoder, if you try that, it puts out a lot of different files. Typically, what you're most interested in is going to be the coding sequences. Those are the cDNA sequences that are described as best candidates. With the eclipsed open reading frames, that is those that are where a reading frame seems to be represented within a larger coding sequence. In that case, the redundancy is removed, and you get a slight reduction in the size of this population versus this population. And otherwise the minimum size, if you're looking at these, Longest means longer than whatever you set as the minimum size. So, for example, if you set 300 bases as your minimum length for transcript decoder, then that will be the minimum size. And anything smaller than that is not going to be presented here. So, that's part of the tool, but it's also helpful to use that information when you're culling your reads. And this peptide here is typically a file, is typically what I use when I go to the annotation stage. Many of you will think about doing BlastX. Don't do it. Don't do it unless you're talking about one or two sequences, because it takes way too long and you waste a lot of time looking at 6 different reading frames when you could be looking at one. The one that looks like a reasonable reading frame in your transcript. So that's all I'm going to say about that today. And otherwise, I appreciate that you guys are good at asking questions, and you can ask more questions, any questions you want at this point, and you always have an opportunity to use the ask form. And I appreciate you using it because it gives you an opportunity to ask questions so that others can benefit, but at the same time you can answer questions yourself. Many of you have learned enough at iPlant that you can also be a great help to other users and save you the time. So,
okay, do people have questions? And again, you can put them in the chat window, or you can unmute yourself and speak.
yes, you can feel free to ask any question you want.
We'll give it a minute in case somebody's typing. Okay, Roger, there's one from Zane again.
Yeah, I'm looking at it. Would you recommend combining assemblies from multiple assemblers and then running CD? You could try that. I've never tried that, and I don't know how it would respond as far as clustering them, but it may be able to do that okay. The other approach is to, and what I've done is to use multiple assemblers or multiple assemblies of different Kamer values, and then use the merge function that you have with the Velvet assembler, and when you're using it with Oasis. And you have to read carefully the manual to use that, but it's helpful. It can be very helpful because you can literally merge assemblies using that using Oasis. It's designed to do that. But CD hit the ESC, I don't know, it may be able to, but it may tend to overload it too.
Okay, the question from Andrew.
Well, okay, Andrew. I like Soapdenovo-Trans because it's an effective and efficient assembler. But if I were doing a project myself, I would probably try Soapdenovo-Trans, and I would try Trinity. But it depends. You know, if I worked in an insect system a lot and I found that my insect assemblies worked better with Soapdenovo-Trans, then I would use Soapdenovo-Trans. Or if they worked roughly the same, I would use Soapdenovo-Trans because I would get my results faster and more assuredly, because Trinity has a bad habit of not finishing sometimes. And it doesn't explain why, and it's not always because it ran out of time, it just doesn't finish. The other thing is that the other questions related to top hat or bow tie or star. Those are top hat is part of and bow tie, which is used with top hat, and maybe star. I don't know. They can be used with reference-based assembly, but not with de novo assembly. Unless again you are essentially doing a reference-based assembly by mapping to something that is close in sequence to what you're trying to assemble. So top hat with cufflinks, using cuff merge for merging. That whole pipeline is great for reference-based assembly, and for a system that's more characterized and that that you study commonly, like Arabidopsis or humans, or as I said before. But if you don't have a reference sequence to start with, then you are doing a de novo assembly, and that's where you really have to use something like a Trinity or Soapdenovo-Trans.
Okay.
And as I said, we do have reference-based assemblers. For example, Velvet, Oasis. I include Velvet when I'm talking about Oases because Oases is sort of the third step that's added to Velvet, Velvet H, Velvet G, in order to do transcriptome assembly. But you can do reference-based assembly with Velvet as a genome assembler, and with Oases as a transcriptome assembly. And you can also combine large reads, like 454 reads, with your Illumina reads using Oases. So, so there are certain advantages to Oases, and Oases is also a good assembler. It's a little more cumbersome to use, and that's why, it may be not the first one that you would reach for. But sometimes, in some systems, it may be the best assembler to use. And, and again, it really depends on what organism, what system you're working with.
Okay, there's a question from Jeannie.
I think generally it's been shown that there are advantages to using paired end sequences for doing assemblies. But I think, there is a law of limiting returns, using a lot of different spacings for transcripts is not as important as it is for genome assembly, because typically your transcripts are going to be shorter one way or another. And again, the emphasis is going to be on being able to assemble a good representative population, but also have some sensitivity. And if you end up, looking at just the larger transcripts produced with your paired ends, you may miss the ones that just aren't lucky enough to catch each side of a paired end sequence and miss it in those smaller sequences that end up as a result. So it depends on what you're doing, but I think generally I think you can say that it's going to give you some advantage in terms of the size of the assembly, but it also will give you some advantage also in the security if you do, because there's a preference to mapping both pairs at once. And if you're getting two independent pairs hitting the same sequence, then and those are both coming up at the same time in your assembly, then I think you're getting more secure information, especially if you're able to fill in that overlap between, or that spacing that was filled in with ends initially. In many cases, you can fill in that spacing if you're only using 500 base pairs spacing, especially for transcripts. So I don't know if that makes sense, but you do add an extra level of insurance, I think, that you are looking at a real mapping. But here again, that is for reference-based assembly. Paired end, you just have the main advantage that you're dealing with larger assemblies in the end.
Okay, Zane, thanks. You've got a suggestion up there. We'll give people a chance to read it. And I can't tell if someone is trying to just speak. Do I hear someone else on the line? Just background noise. Okay.
Yeah, thanks, Zane, pointing out that we do have the, I think, I don't know if Maker is public yet in the discovery environment, but it has been built to work through the discovery environment. I think we're gradually rolling out different sizes of Maker
right.
programs.
In fact, is Shabari still on the call? Shabari looks like he's had to go do something else.
Okay.
So
Yeah, Shabari was working on that.
Yes, Shabari was on the call earlier. He's one of the iPlant scientific analysts, and he's the one getting Maker built within iPlant. And in fact, the next Focus Forum webinar is by Shabari, and it is on Maker. So that's next month. Yeah. So thank you for that that advertisement. Okay, we've got another question here. We're gonna run a little bit long, if that's okay with people. If you
it's up to you at this point.
If you need to go ahead and do that. We understand. And let me tell everyone, I will send out a follow-up email, hopefully before the end of the day today, Pacific Coast time, with the slides and the recording of this webinar. It does take a while for the webinar recording to convert into MP4 format and then get uploaded to YouTube. So that's the hang up. But if you are going to drop off now, do watch for that email. Please give us feedback through the survey and, of course, on Ask. And as Roger said, and I say, you all have a lot of knowledge, so don't hesitate to actually chime in and answer questions on the Ask forum. So, okay, I will be quiet now and let Roger get back to answering C. Esquima's question.
Yes. Well, I think you've sort of answered your own question. I mean, once you have information based on N50 and the sigma, kegma values, and you maybe you've tried some different k-mer settings or different assemblers, you get a sense that you've hit a range that is not necessarily going to get much better. And that's it. I mean, an assembly is a representation of the transcriptome. It's not a perfect mirror of that transcriptome. It's not unless you spend a lot of time going over it and doing multiple sequencing runs that you're going to, and ideally have a reference genome sequence also to work with, that you're going to be able to have a more complete sense of what your transcriptome is. But your assembly is as good a representation as it may be. And I would start working with it at that point. Isoforms. Well, isoforms are those, as you probably know, are the subpopulations for any specific gene, where there are different transcript forms based on different transcription sites being used. And sorting out the true isoforms, that's a tricky thing. And I think you'll ultimately find that unless you have a really complete transcriptome, and that's sort of impossible to get with, a single assembly and in a limited amount of time, you're going to have a better representation of the isoforms for the well-represented transcripts, but not necessarily for the lesser ones. And the reality is, when you're dealing with the question of isoforms and transcriptome assembly, in many ways, your reads come from fragments of RNA sequence ultimately, and many of those reads only fall within one exon, and so those reads only tell you one thing, and that is the sequence of that exon. You may be able to draw information by comparing the representation of different exons as to, well, maybe these, if you have a lot of, say, exon 1 and 3, but 1, 3, 4, and 5 for a gene, but you also have a lesser representation of 2, 4, and 5, then maybe you can say that there are two different isoforms. But it helps to have the junction information, and you do generally get junction information that tells you how many of those junctions you have, and that really tells you how things are linked together better. But in the end, that is the sum total of your information. And you may not be hitting very efficiently some of the outer, the 3' for DNA, for sequencing as it goes, sequences. or exons for an isoform, and so you may not really be able to determine where those, who those exons belong to. Your best representation are going to be the ones that are closest to the poly A, assuming that your RNA started with reverse transcription using poly A. But some of the methods don't do that now, but nonetheless, you're often going to see some skewing in the populations of the exons, and it requires a really good representation of all the isoforms to be able to tease them apart with an assembler, and some of them do it better than others. And that's about all I can say about it. I mean, I think you may be able to do that better by using a higher Kamer value in some cases, but not necessarily, because here again, then you're lowering your representation on the more poorly represented transcripts. Let's see. Oh, okay. Yes, we're going to send you the links.
Oh, so scroll down to the bottom.
Yeah, and then is there an unacceptable thickness for? Well, I think if you're, you can. I think if you're dealing with a eukaryote and you're not getting up in the 90s, then you're not doing real well. Is my feeling, but it really depends on how big the genome is and how many reads you have. That to some extent is dependent on your budget. And, if you want a better representation and a better transcriptome, then you can keep sequencing the same thing using slightly different approaches for a year or two and get a more accurate transcriptome, possibly. But in the end, it's again law of limiting returns and limited budgets, which is more likely the case. And it's not. I think you're going to find that if in many cases with a reasonable start in terms of number of sequencing reads, that you should be able to hit the 90s and the upper 90s ideally. That's my experience anyway. And thanks again for joining us and for the questions.